All right, guys, we're back. We're still in Unit 3.2, Structure and Function of DNA, but today we're moving on to Part 3, the cell cycle and mitosis. All right, you guys ready for this? Now, before we start talking about the cell cycle, remember, in our last video lesson, we were talking about DNA replication, and we said the purpose of DNA replication is so that you have a copy of DNA to put into each cell after the cell divides. So when one cell goes through division, becomes two cells, you need to put a copy of DNA in both of those cells. So if you're going to end up with two cells, that means you need to have two copies of your DNA. But what happens is, before you divide, you have to replicate that DNA. All right, well, now we're done with replication, because again, we covered that in our last video lesson. Today we're going to start talking about the actual cell cycle. Now it's broken up into two main portions. The first part is called interphase. And this is where the cell just kind of grows and does its normal cell job. And then after that is M phase, which is when mitosis or cell division occurs, as well as something called cytokinesis. Now both of those are actually part of cell division, but they both focus on different things in the cell, which we'll get to that when we talk about them. So for the cell cycle, for interphase, there's actually three parts of interphase. So we can take interphase and we can divide it up into three different phases. We have G1, S, and G2. Now G1 is the beginning of interphase. This is what a cell goes through right after it's finished its previous division. It goes straight to G1. And during this time, it kind of checks itself for errors. It'll grow because after division, the cell is small, kind of, itty bitty. So it has to grow to become a normal cell size. It's also going to go through, making copies of its organelles if it didn't do that beforehand, or if it needs new copy or new organelles for whatever reason. And it's also going to go through protein synthesis. And what that means is whatever type of cell it is, whether it's a neuron or a skin cell or a stomach cell, it's going to go through and make whatever protein it's supposed to make to do its job. So for instance, different neurons and nerve cells will be focusing on making different kinds of hormones and neurotransmitters. The cells on the inner lining of your stomach are going to focus on making digestive enzymes. Your skin cells are going to be focused on, working on that sweat or oil or hair. So they're going to be doing whatever their job is in their place as a cell. At some point, they're going to get a signal telling them to start getting ready for replication, or for cell division. And that's when it switches from G1 phase to S phase. So now that we're in S phase, this is when we synthesize our DNA copy. In other words, this is when we take our DNA and we replicate it. We make a copy of it so that we are ready when division occurs. Once we are done replicating our DNA, then the cell switches to G2 phase. And this is all prep for mitosis. So it's going to take in extra material and build extra copies of organelles, such as the centrioles. It's going to take in extra glucose so it can power the entire division process. It's going to kind of build in mass, so it's a good size for when it divides. The daughter cells aren't too itty bitty to function. And once it's done prepping, that's when it switches to M phase, which is mitosis and cytokinesis. So this is kind of showing you, it spends a lot of the time in G1, then it gets a signal, goes to S phase, makes a copy of its DNA, and it goes to G2, preps for mitosis, preps for division, and then it goes to M phase, mitosis and cytokinesis. So daughter cells, the new cells after division, go straight to G1 phase. Now here's the thing. Remember how I said that in G1, the cell checks itself? If it recognizes that something is incorrect, that there's some kind of error that occurred during the division, it can actually remove itself from G1 and go into what's called G naught or G0 phase. We don't necessarily consider this part of interphase because this is a go, no-go step. What it means is the cell will only go to this phase, G naught, if there is an error in it. And if there is an error, and it can be fixed, if the cell is able to correct that error, then it can go back into G1. Then it can re-enter the cell cycle. However, if it can't fix that error, if it's, for whatever reason, it recognizes something is wrong, but it can't fix it, then the cell will stay in G naught and it will actually go through a process called apoptosis, where it will destroy itself. In terms of the body, it's better to destroy the incorrect cells because then you can prevent a disorder from occurring. So if it recognizes, again, that's the key right there. If it recognizes that there is an error, it removes it from the cell cycle and it enters G naught. If it fixes the error, it can go back into the cell cycle, it can return to it. If it can't, then, destroy itself. All right, let's talk a little bit about the chromosomes. So, chromosomes we've already talked about. That is a form the DNA can take, and it's a form that allows it to easily divide itself up and separate itself for division. So before cell division can occur, we replicate the DNA, we make a copy of it, and what occurs when it goes from the chromatin form to the chromosome form, what you actually have are two identical sister chromatids. So although we refer to this as one chromosome, this is actually showing you right here the replicated bit. So if you look, this small arm at the top and this long arm at the bottom, so this left-hand side, that's actually the original chromosome. Well, kind of. This, on the other side, is its duplicated portion. So what happened was the DNA was together, they got separated, replicated, and then they connect. So imagine kind of like this. Yeah, like that. Both of these are pointer fingers, right? They have the joints in the same place. They're roughly the same length. They're connected to each other. So one is a copy of the other, and that's what these are. Now, since they have all the same information, it's just two copies of the same thing, we call them sister chromatids. They are connected by a protein around this region, and this region is called the centromere. So the sister chromatids connect to each other at the area called centromere. This is kind of showing you what we were talking about. Now remember, you have two copies of every chromosome because you get one from mom and one from dad. Now, since they come in pairs, and as we already said in a previous lesson, they have the exact same genes, but they might have different versions of those genes, like eye color. Eye color is going to be on the same chromosome. You get one gene for eye color from mom, one from dad, but there might be different versions. Maybe you get a brown eye allele for the eye color gene from mom, and a blue eye allele in the eye color gene from dad. Now we call this pair homologous chromosomes. Homologous because they are the same. They have the same genes. They might have different versions of those genes, but they have the same genes. Now what happens is during S phase, we replicate those. So here are my replicated chromosomes. And that's what you see here. We have purple and green. Notice how they're individual. Together they are homologous chromosome. We go through and do DNA replication. Notice how we now have an extra version of the purple one. So we had just this one. Now we have a second version of it connected at the centromere. Both of these are considered sister chromatids for the same chromosome. Same with the green. We have this guy here and the copied version here. Both of them are sister chromatids for the same chromosome connected at the centromere. So during G1, your chromosomes would look like this. After S phase, that's when they look like this. So it's just two identical copies of the same genetic section. All right. Now, we've gone through, we've replicated our DNA, we've prepped for division, we are now entering M phase. And the first part of M phase is mitosis. Now, mitosis is a process that divides the nucleus and ensures that each new cell will have a complete set of chromosomes. So whenever we talk about mitosis, we're specifically looking at the separation of the chromosomes as well as of the nucleus. Now, in mitosis, the point of it is to start off with a diploid cell. Diploid simply means it contains both sets of homologous chromosomes. In other words, it's your full set of chromosomes. Remember, 23 pairs or 46 total. That's diploid. So what you want to do is start off with one diploid cell, divide it, and end up with two diploid cells. And ideally, if you did it correctly, then you should end up with two diploid daughter cells that are genetically identical to each other as well as to the original parent cell. Now, there are four parts of mitosis. First part is prophase, then metaphase, then anaphase, then telophase. And kids have used the initials of them to help them keep it in order, keep track of the order. So prophase, metaphase, anaphase, telophase, PMAT, or PMAT as the kids call it. Now, before we talk about what goes on in each phase, I at least want to show you this. So notice here, our original cell has one homologous pair of chromosomes. So one homologous pair, but it's a pair, so there's two individual chromosomes. We have a red and a blue. We replicate the DNA, so now we have their sister chromatids attached. We go through mitosis, we separate the sister chromatids, and we end up with two daughter cells, each with one homologous pair of chromosomes. One red and one blue. One red and one blue. Look, they're identical to each other, and they're identical to our original cell. That's the point of mitosis. Now let's go ahead and start looking at these different phases. We're going to start off with prophase. This is the first part of mitosis. Now in this, the chromosomes become visible. And when we say they become visible, we don't mean that they were necessarily invisible at first. What we mean is that they're switching from the chromatin form, which is so densely packed that you can't see it with a regular light microscope. So we're switching the DNA from the chromatin form to the chromosome form, which chromosomes can be viewed with a light microscope. And with the chromosomes, they are the sister chromatids held together by the centromere. Because remember, we've already replicated our DNA. There's an organelle called the centrioles. They kind of work in pairs, so that's a pair of centrioles, and this is a pair of centrioles. During G2, originally we only had one pair, but during G2 phase, we built an extra pair. And what's going to happen is these centrioles are going to start moving to opposite ends of the cell. So they're going to start separating and moving to the opposite poles. As they are moving, they're going to start building these fibers called spindle fibers. Now, we want to move and separate our chromosomes. That means that they can't be bound in their protective nucleus sac. So we have to get rid of the nuclear envelope. We have to free the chromosomes into the cytoplasm if we want to be able to move them around. So the nuclear envelope is going to start disappearing and breaking down. Don't worry, we're going to rebuild it later. But that's going to free these chromosomes to the cytoplasm, where they can then start attaching to the spindle fibers. But we'll talk about that in the next phase. So the spindle fibers are actually going to attach to the chromosomes. Specifically, they're going to attach at the centromere. When they attach at the centromere, then they're going to start pulling and maneuvering the chromosomes so they line up along the center of the cell. Now they're center in terms of between the opposite centrioles. So they're in the middle of the cell and the centrioles are on opposite ends. The centrioles build the spindle fibers. The spindle fibers attach to the centromere of the chromosomes and then help line the chromosomes to line up along the middle of the cell. That takes us to anaphase. The centromeres are going to split and separate the sister chromatids. Now, the reason why this occurs is because those centrioles are going to constrict and contract and pull on the spindle fibers. That's going to cause the spindle fibers to pull on the centromeres of the chromosomes, causing them to split. Well, we've now separated the sister chromatids, and the spindle fibers are going to keep pulling on them and dragging those sister chromatids to opposite sides. So here we split the centromere and separated the two yellow sister chromatids. We separated the blue sister chromatids. We separated the red sister chromatids. We separated the green sister chromatids. And now we're dragging them to opposite ends so they are closer to the centrioles. And that brings us to telophase. Telophase, remember, in anaphase, we drag the sister chromatids to opposite ends of the cell. Well, now that we have them grouped at opposite ends of the cell, we're actually going to rebuild the nuclear envelope around them. So since there are two groups of the sister chromatids, we are going to form two new nuclei, one around each group, as you can see here. We are reforming the nuclear membrane around each group. The chromosomes are going to disappear. Again, doesn't mean that they're like poof, gone like magic. It means that they're going to go back into that super highly condensed version known as chromatin. So they're going to get all back together, wind themselves back up, back into chromatin form. And the spindle fibers are going to start breaking down. We don't need them anymore. They did their job. So we are going to start breaking them down. And as you can see again, the nuclear envelope reforms around the group of chromosomes. They're going to go back into their chromatin format. Notice the spindle fibers aren't as visible because we're breaking them down. That is it for telophase. Now we have not finished separating the cell because that is, ta-da, cytokinesis' job. Now cytokinesis is not part of mitosis. Mitosis deals with the chromosomes and the nucleus. Cytokinesis is everything else, the rest of the cell. Cyto refers to cell. Now, this can occur at the same time as telophase, but it itself is not part of telophase, and it itself is not part of mitosis. So what it is, we have to now divide the cytoplasm, the organelles, and pinch in the cell membrane in order to separate into two new daughter cells. Because remember here, it's all still connected as one cell. But we want two cells. So what happens is the membrane, notice how it's kind of pinching inward on either side between the two nuclei. So it's going to keep pinching inward and inward. And as it does that, that causes the cytoplasm to stream to both ends of the cell and bringing with it all the other organelles that are in there. So the mitochondria are going to move to opposite ends. Remember, we also made extra copies of our organelles during interphase. We're going to separate those extra copies and move them to opposite ends of the cell. The cell membrane is going to keep pinching in and pinching in and pinching in until it fuses. It's going to touch and fuse, and at that point, So we have our cell like this. Cell membrane pinches in, pinches in, pinches in, fuses, and then after it fuses, it pops apart. Hold on one moment. Let me pause this. All right, sorry about that. There were someone at our front door. Anyways, so they pinch in, so they connect, fuse, and then pop apart. All right, so you guys ready to kind of put all this together? Take a look at the overall picture. So here we have interphase. We have the chromatin, centrioles, etc. During this part is when DNA replication would occur. So here we are seeing prophase. Now, how the chromosomes would have looked before replication would be one long pink, one long green, that's one homologous pair, one small pink, one small green, that's another homologous pair. So we have two homologous pairs, 4 total chromosomes. Now we've replicated our DNA and we can see the sister chromatids attached to each other via the centromere. Prophase, you can see the chromosomes, they become visible. The nuclear membrane has disappeared, it's broken down. The centrioles are also starting to move. And at this point, they're also going to start forming those spindle fibers. And that takes us to metaphase. The spindle fibers have attached to the centromeres of the chromosomes and helped maneuver them so they line up in the center of the cell between the centrioles. Anaphase, the centrioles pull on the spindle fibers, which then pull on the chromosomes, causing the centromere to split, separating the sister chromatids and pulling them to opposite ends. Then we have telophase, where we have our chromosomes. They're going to go back into chromatin format. We've reformed the nuclear membranes, and we've broken down the spindle fibers. This is also the point where cytokinesis will occur, and that's the pinching in of the membrane, causing cytoplasm and organelles to stream to either end, so that it will eventually pinch all the way in and whoop, pop apart to form 2 cells. Now remember, here's what we started with. And before replication, before DNA replication, we said it was one long pink, one long green, one small pink, one small green. Hey, look! One long pink, one long green, one small pink, one small green. And when those, after end phase, mitosis and cytokinesis is complete, both of these daughter cells will enter G1 and start doing their normal cell jobs. When they get to S phase and replicate their DNA, then they're going to look just like this one here. That's why we call it the cell cycle, because it keeps going through it over and over. Now, different types of cells, I will say this, different types of cells go through the cell cycle at different lengths of time. For instance, your skin cells and like the cells on your tongue and all that, they can, go through the cell cycle within a matter of days. There are other cells, like your brain cells, that will go years in between cell cycles because they don't really need to replicate that often. So they've been programmed, when they were told to be brain cells, to not go through cell division as often. So different types of cells have different timelines with which they divide them. All right, and here's another picture. So this is right here. So here we have our cell in G1 of interphase. Notice long purple, long red, short purple, short red, goes through replication, and we end up with our sister chromatids attached. Now here we have prophase. Nuclear membrane is going to disappear, chromosomes are visible, centrioles start to move, and they start forming their spindle fibers. We have metaphase. These spindle fibers attach to the centromeres of the chromosomes and helps to line them up along the center of the cell. Spindle fibers pull on the centromeres, or splitting them and separating the sister chromatids, and will actually pull the sister chromatids to opposite ends of the cell. Then that takes us to telophase, where the nuclear membranes will reform around the groups of chromosomes. The chromosomes will go back into chromatin format, and the spindle fibers will break down. At this same time, cytokinesis is going to occur. See the membrane pinching in, causing the cytoplasm and organelles to stream to the two ends. Eventually, that cell membrane will pinch all the way in, fuse, and pop apart, and we are left with two daughter cells. And look at that. Long purple, long red, short purple, short red. Long purple, long red, short purple, short red. Identical to each other and identical to the original parent. All right, now we have some additional resources to help you guys out with this. Science in Pajamas, we have one on the cell cycle. We're just kind of discussing that, the different parts of it, what occurs in each one, each part. And we also have chromosomes. We talk about chromosomes as well. Their structure, diploid versus haploid, all that fun stuff. And then for other videos, we have creative learning, a mitosis 3D animation. It kind of just runs through everything, gives you an idea of it. Amoeba Sisters, Mitosis, the amazing cell process. The Amoeba Sisters, the cell cycle and cancer. And then we have Bozeman Science, cell cycle, mitosis, and meiosis. And then Don't Memorize, Introduction to Mitosis. These will be good resources to help kind of reinforce what we've talked about, help explain it in different ways to ensure you understand it. And if you have questions, you can also feel free to email me, get me through our Google Classroom or in class, all that fun stuff. I will post a link for this though in the video description so you have access to those additional resources. All right, well, in the meantime, you guys, stay awesome, stay curious, stay safe and healthy. All right, guys, you take care of yourselves. Bye-bye.